Welcome to The Explainer. Today, we're diving straight into the fascinating, totally invisible war that humanity wages against microorganisms every single day. I mean, we are constantly surrounded by this microscopic world, and our ability to control it is, quite literally, the foundation of modern human survival. It's a high-stakes battle of science, heat, chemicals, and math, and today we're going to break down exactly how we fight it.
There's this fantastic quote by Dr. Harry Burns that perfectly sets the stage for what we're talking about today. He points out that understanding germ theory and sanitation has actually done more to extend our life expectancy than any other medical innovation in history. Think about that. Before we had fancy antibiotics, way before complex surgeries, we had to just figure out how to stop these invisible killers from spreading in the first place.
To really put a human face on this threat, we have to talk about Mary Mellon. You probably know her by her infamous nickname, Typhoid Mary. Back in the early 20th century, Mary was an asymptomatic carrier of Salmonella typhi. She felt totally fine, but because she was a cook and because hygiene practices back then were, well, not great, she unknowingly spread a deadly disease to the family she worked for. She was often dubbed the most dangerous woman in America.
But here's a really crazy twist. As infamous as Mary was, causing 10 outbreaks that led to 53 cases and 3 tragic deaths, she actually wasn't the deadliest asymptomatic carrier of her era. Not even close. There was a lesser-known cowman and milker from the UK, recorded simply as Mr. N, who was far deadlier. Because his unwashed hands interacted with milk, which went out to a much wider community, his poor hygiene resulted in 14 outbreaks, causing a staggering 207 cases and 43 deaths.
And honestly, that brilliantly illustrates why our absolute first line of defense is shockingly simple. We just need to wash our hands. Following the World Health Organization's hand-washing steps mechanically removes these dangerous pathogens right off our skin. And no, a quick 2-second splash doesn't cut it. The science actually dictates a 40 to 60-second process of wetting, applying soap, really rubbing palm to palm to detach those microbes, rinsing, and drying with a single-use towel. That simple mechanical action is the absolute bedrock of public health.
Section 1. The Rules of Engagement, Defining Our Arsenal.
First up, we have our absolute highest standard, which is sterilization. Think of this as the nuclear option. When something is sterilized, it means the complete and total destruction of every single microorganism and virus present. Even highly resistant bacterial endospores are wiped out. Nothing survives. This is the standard we use for things like surgical tools and commercial canned food.
Next down the ladder, we have disinfection. This is a slightly scaled-back approach. It guarantees the destruction of most microorganisms and viruses, specifically the pathogenic ones that actually make us sick. But crucially, disinfection applies exclusively to non-living things: countertops, lab benches, medical equipment. The chemicals we use here, like strong aldehydes, are just way too harsh to ever put on human skin.
And finally, there's antisepsis. Functionally, this is very similar to disinfection. The goal is still a massive reduction in pathogens, but it's specifically formulated to be safe enough to use on living human tissue. So, when a doctor preps your arm with iodine or alcohol before a shot, they aren't sterilizing your skin and they aren't disinfecting it. They're applying an antiseptic. Section 2. Cellular Sabotage. How microbes actually die.
So how do we actually pull this off? Well, we generally use 2 main tactical approaches to break a microbe down. The first is targeting their proteins using extreme heat or changes in pH. This literally forces the microbe's precisely folded internal proteins to unfold, a process called denaturation. Once that shape changes, the enzyme is completely dead. The second method is a chemical attack using surfactants, which are found in basic soaps. These lodge right into the cell membrane and literally blow holes in the cell's armor, causing its vital insides to leak out, dead instantly.
Or we can bypass those outer defenses entirely and go straight for the microbe's genetic code, using radiant energy. Think about how UV light operates. It sneaks right past the cell membrane and gets absorbed directly into the DNA. This causes adjacent thymine molecules on the DNA strand to awkwardly link up with each other. It kinks the whole double helix, causes breakages, and stops the cell from ever replicating again.
Game over.
Section 3. Heavy Artillery, the Math of Eradication.
When we need absolute sterilization using dry heat, we do not mess around. Take infrared incinerators, which are super common in microbiology labs. They safely blast tools with heat, reaching 800 degrees Celsius to burn off any biological material. And on an industrial scale,
To safely dispose of highly infected materials, massive furnaces operate at an astonishing 6,500 degrees Celsius, turning microbes into pure ash.
But heat isn't just about turning things to ash, it's also about really precise mathematical control, especially in the food industry. To understand the stakes here, just consider this massive number, 1 trillion. That represents a typical starting population of deadly Clostridium botulinum spores you might find in a commercial batch of canned food before processing. If even a few of those highly resistant spores survive, they produce a toxin that causes botulism, which is a fatal paralytic illness.
To beat those odds, food scientists use a calculation called the D-value, or decimal reduction time, to conquer that 1 trillion. The D-value is the exact amount of time it takes at a specific temperature to kill exactly 90% of the microbial population. Now, because microbial death is logarithmic, you can never technically hit absolute zero. Instead, the goal is to apply heat over and over, dropping the population by 90%, then 90% of what's left, then 90% of that, until those 1 trillion deadly spores are reduced down to a statistical probability of just one single spore remaining.
Section 4. Safe Food and Milk, the Refrigerator Frontline.
Let's look at how we apply this to something as everyday as milk. Standard pasteurization uses a relatively brief, controlled heating process. It kills the dangerous pathogens that make us sick, but it intentionally leaves harmless spoilage microbes behind. That's why your regular milk still needs to stay in the fridge and goes bad after a couple of weeks. But then there's ultra-high temperature, or UHT sterilization. This blasts the milk at a blistering 140 to 150 degrees Celsius for just 1 to 3 seconds. It totally sterilizes the liquid, which is exactly why you can buy those boxes of organic milk that just sit perfectly safe in a warm pantry for months without spoiling.
Now, for larger volumes of liquids, or for reusable hospital tools that need total sterilization without, being incinerated to ash, we rely on the total workhorse of microbiology, the autoclave. It brilliantly traps steam inside a heavy metal chamber. By forcing that steam under high pressure, the laws of physics allow the temperature to rise past the normal boiling point, hitting a lethal 121 degrees Celsius. This pressurized moist heat aggressively penetrates materials and denatures microbial proteins way more efficiently than dry heat ever could.
Section 5. The Global Sanitation Mission, WASH.
Despite all our incredible mastery over UHT processing and infrared incineration, there is a very sobering reality we have to face. According to data tracking Sustainable Development Goals, as recently as 2015, a staggering 2.4 billion people, that's about 1 in 3 humans on Earth, still lacked access to basic sanitation facilities. On top of that, over 663 million people did not have access to safe, clean drinking water. They are essentially fighting this invisible war completely unarmed.
And the devastating consequence of this gap in basic WASH initiatives, which stands for water, sanitation, and hygiene, can be summed up in one heartbreaking number: 700,000. That is the number of child deaths that occur annually simply from diarrheal diseases. Diseases that are entirely preventable if the basic hygiene principles we discussed at the very beginning of this explainer were just universally accessible.
Which leaves us with a massive question to end today's explainer on. We know exactly how to defeat these pathogens. We understand their DNA, their cell walls, and the exact math required to eradicate them. So if we possess the science and the technology to completely conquer the microbial world, how do we bridge that final gap to ensure this basic, life-saving infrastructure reaches every single person on Earth? It's a question of human priority and one we absolutely have to solve. Thank you so much for joining me on this journey through the invisible war. Keep asking questions, and I'll catch you in the next explainer.